I'm Julia Douthart with Fierce Biotech, and I am delighted to welcome today's speakers. Ian Chan, who is the CEO of AbPro, and Ruben Zhang, Director of Antibody Process Development and MSAT at GeneScript ProBio. You can read their full bios on the left side of your window by selecting the Speakers tab. Quick technical note or two before we begin. This webcast is being streamed through your computer. There's no dial-in number, so for best audio quality, please turn your computer speakers up. This webinar is being recorded and will be available on demand within 24 hours after the event. Today we'll have presentation and then we'll go on to your questions from the audience. So please submit your questions using the questions and answers tab on the left side of your screen, and you can do that at any time during the webinar. Okay, I think we're ready. Ian, please go ahead.
Thank you very much, Julia. It's great to be here today. Today we're going to give a quick overview of our bispecific platform. AbPro is a Boston area biotech company focused on next generation antibody therapies for people in medical need. We're focused on a couple of different areas: immuno-oncology, infectious disease, and also eye care. And our platform, which includes the diverse immune platform and also our multi-mAb, which is the bispecific platform, which we'll go through today, has a number of features that make it highly advantageous for treating patients in need. So I'm going to go through the multi-mAb platform today. So that's our bispecific platform, which is proprietary to AbPro, which we've been developing for the last several years. And it's going to be a short overview. And then, of course, we can welcome any kind of questions that you may all have. So bispecifics are a logical evolution of antibody therapies. They've been in use, and now there are a number of approvals. With our antibody platform, there are a couple of ways that we approach it. Firstly, we're able to make all the components of bispecifics using our diverse immune platform. So the components are single monoclonal antibodies, which we create using the platform. And these are generally antibodies against traditionally very, very difficult targets, which the company has had multiple years of experience creating, almost 300 different campaigns for almost, I would say, all major pharmaceutical companies, biotechs, academic labs. So once these individual components are created, we combine them into bispecifics. So there's the ability to really start from the beginning of the entire process, so building the Legos in-house and then reassembling them into bispecifics with various different formats against almost all major targets are possible using this platform. So the evolution of the field has been actually quite fascinating. So bispecifics initially were not full antibodies, but were actually fragments. So these had, I would say, initially very, very interesting applications. So I'm referring to the initial platforms, the CD19, CD3s that got approved, primarily BiTEs that were generally effective, but had short half-lives. So these were groundbreaking in the fact that they got approved, but certainly had drawbacks in the half-life. And then the field has moved on to generating different formats that can now have longer half-lives and with different therapeutic windows. So these have been very interesting as well. So some of these have now advanced very deep into the clinic. Where we're different is the ability to have a long half-life, but also a very, very broad therapeutic window. So that's our proprietary tetrabyte format, which we have now a number of molecules that we're advancing into the clinic. So the main advantage here is a very broad therapeutic window, meaning the ability to see efficacy before toxicity, and then also the ability to have a long half-life. So that's the main advantage of our platform here. So here we show the different the evolution of the whole field. So the first generations, they didn't have any bivalent binding. The circulating half-life was very short, so people had to wear a continuous pump to really infuse drug. Although they were generally safe, but the dosing had to be very carefully modulated. The second generation had better features, but really the manufacturing was really continually, the yields were generally not as high, primarily because the mismatch of the two different antibody chains was generally difficult to pair up. And so there was a lot of yield that was lost during the manufacturing process. So our approach has been to really overcome all these disadvantages in our platform, and we believe we have. So there's not only bivalent binding to a tumor, long circulating half-life, it's minimizing CRS, generally low risk of immunogenicity since these are natural antibodies, and also the manufacturing the yields are higher just because of the symmetry of the molecules. So we've basically taken this approach and scaled it up. So there are a number of molecules that we have developed all in-house. They're against some of the biggest unmet medical needs that we see. So HER2, CD3, GPC3 for liver cancer. We also have a Claudin 18.2 for gastric cancer, which was one of the most, I would say, difficult antibodies to develop in the industry, which we were able to create using our diverse immune platform. So this is a highly scalable approach that we can simply go after tumor targets on one hand, combine it with various immune cells, and we can do all this in-house, and then create novel bispecific antibodies that are not only, we believe, are going to have a better safety profile, but also very nice efficacy, and then also highly manufacturable. And then T-cell engagers are actually it's a very interesting topic right now, just because it takes the same approach as a CAR T, but is actually a lot more manufacturable. So CAR Ts proved that this approach can work, but where it got stuck was really on the manufacturing side. So they had to be really custom made for patients. Bispecifics really allow a broad manufacturing, one size fits all type approach that CAR Ts does not have. And then our platform is actually natural. So we use the most proven type of technologies, which are really in vivo derived antibodies to start with. So these generally have very low immunogenicity, and then we can basically go after targets of interest and then recombine them with any type of immune cell that's actually interesting and effective. So this is a highly proven technology. Multiple antibodies have been generated this way and has been proven to be safe. And antibodies are the largest class of approved molecules in the biotechnology field. So the manufacturing generally is, well, I would say straightforward. And the yields are also very, very good. So that's why we actually adopted this approach. And of course, our diverse immune platform has specific advantages to be able to create these much faster and also against traditionally difficult targets. So that's an overview of our multi-map platform. So at this point, I can turn it over to it looks like GenScript would be next here.
My name is Ruben. I'm from GenScript ProBio, and it's my pleasure to share some of our experience regarding bispecific CMC strategies. Today, we will go through a quick introduction about GenScript ProBio. Then I will introduce our GS host cell system. We will go a little bit about developability assessment, but last but not least, we will go a whole story about the CMC strategy for bispecific antibody. A little bit about GenScript ProBio. So we have office or cell office or manufacturing site all over the world. In China, we have our headquarters, and the process development group, clinical manufacturing site 1 and 2 are located in Nanjing. And in Zhenjiang, we have our plasmid and virus process development. And also, we're building our biologics clinical 3 and commercial manufacturing in Zhenjiang site. And in New Jersey, we have our plasmid and virus vector GMP manufacturing facility in New Jersey. And also, we have our GenScript ProBio headquarters in New Jersey for North America. And for Europe, we have headquarters in Amsterdam. And in Singapore and Tokyo, we also have our own cell office or headquarters. On the bottom are the selected companies that collaborated with ProBio before. We definitely have a fast-growing track record. If today we're going to talk about more on CMC side, so if you look at the right, we have more than 110 cell line projects for IND filing. And so far, we have went through 71 CMC and CMO projects. Besides those projects, 40% of the projects are from United States, Europe, and Asia Pacific. For now, we got 19 global INDs, and we altogether manufactured GMP batches about 77, and we went through 43 customer audits, and there was no critical and major matter was found. The entire GMP facility are operated under FDA, EMA, and NMPA standard, and we passed EUQP last year. In terms of the modality, 46% of all ProBio's CMC projects are non-mAb projects. We have 19% of the projects are bispecific or trispecific product, and 20% of our projects are protein-related projects. And altogether, 54% of the projects are traditional mAb project. For the process development for bispecific antibody, currently we have 5 projects delivered, and if you look at the bottom, they are the modalities we worked with. There are SFV fused with mAb, in vitro assembly ones, and the classic knop into whole. And also we worked with projects that needs to connect the cyclotine to the mAb. And also there are the novel molecules to connect SCFV together. So far, we have a 2,600 liter GMP manufacturing capacity. And if you look at the right side of the slides, we have our tox batch manufacturing plant that has 200 sativa bioreactors, and in biomanufacturing center we have a 200 liter bioreactor and 2 500 liter bioreactors. By the end of this year, we will have fill and finish capability, which means we can do DS and DP in the same site. Currently, the instruments are in the qualification process. And by the end of next year, we will have our commercial manufacturing center, which have 8 2,000-liter bioreactor, and in the same center, we will have a drug product capability as well. I want to talk a little bit about our cell line because this is where the projects start. And if we have a good cell line, if you have a good host cell line, the project will be much easier considering the product quality and also considering the titer. A good cell line or a good clone is always a good start for the project. From GenScript ProBio, we have 2 cell line. First one is CHOK1 genes, which is a wild type CHOK1 from ECACC. And also we modify the CHOK1 genes. So this specific cell line with ADCC plus that can produce afucosylated antibody to increase the ADCC effect. And our host cell line have a clear legal documents to make sure to guarantee the success of the IND filing. To couple with the host cell line, we also have our own in-house vectors. For bispecific antibody or monoclonal antibody, we have a pGene-HT 1.0 DGV or 1.0 up. These are both for monoclonal antibodies. DGV was a classic vectors we have been using all the time. And the last year, we built our new vectors, which is called 1.0 up. On average, it will have more 20 or 30% of the more titer compared with the DGV vector. And for the product that is protein, sometimes it's hard to express in the cell. We also have the 2 G-Unic vector, which were licensed from other company. Using this vector can significantly increase the titer for protein drugs. Besides the host cell line and vector, ProBio also have its clone screening system. It can enable us to do clone screening efficiently and within applicable to compliance. In the same time, is high throughput, so we can select more clone, which means we can have a higher titer clone in the future. For cell line development, so I want to show some data which is summarized from our past project. So if you look at the left side, for monoclonal antibody, majority of our project after process development in classic fed batch process, we will have 5 to 7 gram per liter as the final titer. And even more than 10% of the project reached more than 7 gram per liter at the end of the process development. And now of the project, the final titer reached below 3 gram per liter. On the right is showing the host cell line's performance with different molecule modalities. For monoclonal antibody, on average, the titer is 5.48 gram per liter. And for bispecific antibody, the average titer is 3.47 gram per liter. Here, I want to say this bispecific antibody is limited to the FC region containing product. We have 3.47 gram per liter. And for the ones without FC region, is case by case. And for protein or enzyme product, we have 2.16 gram per liter on average. This slide is showing a quick study about ADCC enhanced cell lines performance. As you can see, for the ADCC, between the 2 cell lines, they have similar viability performance, and they also have a similar viable cell density performance. And the end of the titer without any process development, both of them reached more than 3 gram per liter. This slide further shows how this performance of the ADCC enhanced cell line regarding the N-glycan profile. If you look at the graph on the top, there was no G0F at all, which means the field oscillation was completely limited to a very low level. And for the normal GNS cell line, the G0F is as a normal level, which is around 90, 95 or 96%. So field oscillation was greatly reduced by knocking out the FUT8 sequence. This further shows the activity of the ADCC activity with a product which produced from the ADCC enhanced cell line. Previously, we just showed the change of the N-glycan. And in this slide, we want to further verify the functionality in different potency assays. The first assay is an affinity assay that binds to CD16A. You can see the affinity was increased by 3 to 4 times. And in the middle is showing that the activity of the ADCC activity of fucosylated antibody was increased by 10-fold. And on the right graph, you can see the EC50 was reduced by 6 times. After the cell line, a lot of clients, there is a common questions the clients would ask is, I have a different candidate A, B, C, or even 4 or 5 candidates. They have a similar function and they have a similar pharmacology performance. However, which one I should pick in order to start our CMC project? For these kinds of questions, usually we would recommend to do developability assessment. What we do is we use the host cell line and vector system of the cell pool to generate this developability material. Then this material will put into 3 extreme conditions, like a high temperature, low pH, and a freeze-thaw process to test its developability. For high temperature one, we want to test if this drug candidate can be stored in room temperature, the stability of the storage. And for low pH one, we want to make sure that this candidate can go through the purification without any major concern. And for freeze-thaw, because the drug substance will be freezed and thawed during the production and also the shipments. So we want to make sure this candidate is okay to go through all these harsh conditions. On the right, I'm showing a good example of 3 candidates. For the candidate A, candidate B, obviously they have some quality issue during this developability assessment. So to us, the candidate C would be the best one for the CMC development. Next, let's talk about the CMC strategy for MAP and bispecific antibody. This is a quick overview regarding ProBio's capacity and the timeline design for monoclonal antibody and bispecific antibody. For monoclonal antibody, the key consideration is a fast timeline, and we want to keep this process cost efficiency. The CMC strategy, most of the time, we would recommend to do fast CMC, and we would use fed batch process. Usually, these are applicable to IgG1 and IgG5 and IgG4, and they will be finally formulated into the liquid form. From DNA to tox, it takes 6 months, and usually from DNA to IND, it takes 12 months. The reason I label the DNA to tox time here is because for most of the clients and most of the project, from DNA to tox are the real limiting step for IND application. After the delivery of the tox material, the critical timeline becomes to the tox study. And as the early we can get the tox material, the earlier we can apply for the IND, because tox study takes almost 6 months of the time. And during the tox study time, ProBio will produce the GMP batch and getting stability study ready, and also getting all the IND application materials ready. For bispecific antibody, because depending on the design, sometimes it's a little bit more complicated compared with the map, because during the bispecific antibody manufacturing, we would have a lot of a byproduct clearance problem. Sometimes it's a half antibody, sometimes it's a homodimer, and sometimes it's aggregation. So for this type of projects, we wanted to develop a fully optimized process to achieve high productivity and purity. Cultivation mode, we use fed batch or intensified fed batch. This really depending on the specific scenario. If clients really need a high titer in order to support the future commercial plan, we would use intensified fed batch, which can double the productivity in short period of time without impacting the product quality attributes. For this bispecific antibody platform, it applies to different formats of bispecific antibody. And from DNA to tox, it takes 1 more month compared with the monoclonal antibody. For the 7 months, this format only limits to symmetric bispecific antibody. If we have a symmetric bispecific antibody, it takes 1 more month, which means it's 8 months from DNA to tox. This slide is showing a typical timeline for map fast CMC. You can see at the end of month 6, the tox batch will be delivered, and at the beginning of month 8, the GMP batch will be delivered. Compared with the fast CMC, the bispecific product most of the time will have 1 more month for purification process development and confirmation. So the tox batch will be delivered at the end of month 8. Now I want to switch the gear. Let's talk about the challenges for bispecific antibody CMC. During the cell line development, you may have challenges, for example, low titer. And for process development, you may have problem like a stability problem from this specific project. And during the purification, you may have a problem of byproduct and impurities. And last but not least, for analytical development, bispecific is also something different from monoclonal antibody in terms of how to measure the byproduct, how to measure the impurities, and even how to verify its potency in vitro. So how do we solve those pain points? For cell line development, we modulate the polypeptides by optimizing the plasmid ratio to reduce the mismatch and improve the titer. For cell culture development, still we use high throughput screening strategy that we can pick the best media compatible for specific projects. And we would optimize the certain parameters in the process. And last but not least, we will try to do intensified fed batch if we really need a high titer. For purification, we would to use a multi-model resins based on the type of the impurities. And also in downstream, our scientists would spend quite some time for the process parameter optimization in order to get the best purity and highest recovery rate. And for formulation development, it's more similar to what we typically do in monoclonal antibody. The only difference is we would recommend to use lyophilization if the specific projects have a stability issue. For bioassay, we have developed platform methods to detect the homodimer. And also we have our own cell engineering group that can support our bioassay group to design the functional bioassay based on the mechanism of the action. First is the cell line development. if this this is a bispecific antibody, we would code the target chain in different vector. We would use the 2-vector system. Then we would adjust the vectors ratio. By doing this, it allows us to have the best titer and the lowest impurities. We can solve this problem in the clone selection area. There are several projects we applied this strategy, and after you find the right vector ratio, your titer and the impurity level will be both optimized. And for now, like I said, for bispecific antibody, our average titer is 3.45 gram per liter. For cell culture development, it's more similar to what traditional MAP process would do. We would optimize temperature, pH, agitation, control PCO2 osmolarity, and also depending on the product activity and also product quality attributes, we would decide the culture duration. For culture mode, we usually use fed batch process. Again, if we need high titer, we would do intensified fed batch. It will double the titer in a short period of time. For product that is truly unstable in a cell culture environment, we would apply perfusion process. By using this process, we can exchange the product out of the bioreactor immediately, and this product can be kept in 2 to 8 in cold environment to guarantee its storage and stability. For cell culture development, I'm just showing a quick example how would those factors works. For the up left, you can see after we supplement enough amino acid, immediately the titer was increased. And on the upper right, you can see the effect of the pH. Like when we apply different pH range, definitely it helps to optimize the titer. And for high density inoculation, which is the intensified fed batch, you can see using the intensified fed batch, it will almost double the titer, and for every case. For temperature shift, some of the projects they prefer with temperature shift, and some of the projects just constantly use 37-degree and throughout the process. How to improve the purity in different ways? If this product is truly unstable in the cell culture environment, you can see on the left graph, during the fed batch process, there is only 0.12% intact product. And using the perfusion mode, we got almost 70% intact product. And for on the right, you can see as the culture duration goes on day 15, there are aggregation growing. So in this case, we would shrink our culture time to 14 days or 13 days to guarantee the product quality. For N-glycan modification is really depending on the specific scenario. Depending on the need, we would add trace element or special amino acid in order to modify the N-glycan. On the right, if we have a project for a biosimilar, the first thing we would do is to screen different cell culture media so we can find the best formulation that fit the target product. For purification, we have different formats to deal with different issues. For aggregation, fragment, or mismatch, we have different chromatography we can use. And which is one more thing is important for purification is we also have our different ways to detect the byproduct. We may use the difference in affinity to detect our byproduct, and also different in charge or different in hydrophobicity. This is a good example of using the multi-model resin. Compared with the traditional resin, multi-model resin give us better recovery rate and also better purity after each step. Take on the left graph, you can see using a traditional cation exchange column, the recovery rate was only 15%, and using the multi-model column, the recovery rate increased to 70%, and another multi-model column, it increased, the recovery rate increased to 76%. And if you test the purity of the product, the multi-model resins with a much higher recovery rate, they still maintain a very good purity. On the right is another example of using the multi-model column. It can significantly decrease the homodimers. And on the right, you can see the product purity is 99% using multi-model resin, and the homodimer resin homodimer content was decreased to less than 1%. For formulation development, I would say this is similar to what we do for monoclonal antibody. So I wouldn't spend too much time on this slides. One more thing is if we have truly unstable product, we would use lyophilization instead of the liquid form. For analytical development, we would develop different methods to detect the impurities depending on the specific scenario. If the impurity have a different isoelectric point, we would use ICEF. And if the impurity have a different molecule weight, we would use SDS page. And for sure, we have our LCMS. It's always a good way to monitor the undesired pairings. If the homodimer and the target are similar to each other, and sometimes it cannot be detected by other ways, LCMS is always a good way. For bispecific antibody, another big problem, I would not say it's a problem, but another point that people have to pay attention is how to verify its potency or bioactivity. And this is still depending on the specific scenario. If this bispecific antibody, they have two targets, but the two targets are both immunomodulars, then we would carry out two different cell-based assay to separately assess the potency of each antigen target arm of the bispecific antibody. This is for example, we may have a bioassay 1 for target A and a bioassay 2 for target B. However, if this bispecific antibody is a T-cell engager, then we would do a dual binding ELISA to assess the binding activity of both antigen, and we would develop a cell-based assay to assess the T-cell activation. So this is the two different types of the bioassay was developed for the specific need of this product. Again, we have our GenScript ProBio have our own cell engineering groups that can generate the cell line for bioassay based on their needs. In the table, I'm showing some example and the example targets for different categories. The categories including cell bridging, bispecific antibody, dual target blockade, and or dual immunomodulars. Here I'm going to do a quick case study, and this product is a trispecific antibody, and we got the FDA and NMPA IND clearance for this product. Something I'm going to go over quickly. So by doing the media screening and amino acid supplement, we successfully improved the titer by 62%, and we add manganese to lower the MEN5. And during the purification development, the purity was improved from 84.7%, which is a platform performance. And after the optimization, the purity was increased to 98.6%. And we also developed the suitable bioassay to detect the potency of the multiple targets. This is a tri-specific. These are the actual data. If we assume from the platform, the clone selection titer is 1. After media screening, the titer was increased to 1.36, and after the parameter optimization, the final titer was 62% higher compared with the platform titer. And during the process development, we also noticed MEN5 is higher than normal, and most of the time we want to control the MEN5 under 10. So we add the manganese to modify the N-glycan, and you can see after the modification, which is a blue column, the MEN5 content is well below 10%. For purification development, there are not too much trick. What we did is first we do resin screening. After that, we may have to optimize elution parameter, washing buffer, and optimize the loading capacity. Bioassays development is interesting for this specific project. So for tri-specific, the target 1 or target 2 are the marker of the B cell, and the target 3 are the T cell co-receptor. So our bioassay group was designed 2 bioassay. First bioassay will verify the activity of target 1 and target 3. And the second bioassay have to verify the bioactivity of target 2 and target 3. Another good, interesting case is the in vitro assemble of a bispecific. So this is a non-FC containing bispecific product. It only has the scFv part. So we expressed the scFv part separately in different culture process and purify them. After that, both of the scFv part was conjugated with a linker, and the final step was to conjugate both scFv part. We was adjusting the different ratio of both scFv1 and scFv2 by optimizing the ratio. We significantly increased the recovery rate from 30% to 64%. In conclusion, so ProBio have a leading bispecific antibody CMC platform, which can break through many CMC barriers. We have a high expression system, we have a good yield and a strong activity assay group. ProBio's CHOKI-1 gens and the CHOKI-1 ADCC system can provide its clients world-class productivity and ADCC enhancement, like I showed in the previous slides. And ProBio has a well-established bioassay platform that can accurately measure the target MOA for virus bispecific antibodies. With this, I will finish my today's presentation. Thank you very much for everyone's attention, and now questions are welcome. Thank you.
Excellent, thank you. We do have some questions for both of you, actually. So let's go ahead and move on to the Q&A portion of the program. If you have questions during any of the webinar from now until closeout, you can go ahead and put those in the queue. If for some reason we can't address them now, if you've given us permissions, we'll go ahead and connect with you after the event. So you will get your questions answered. So I am going to start here with a question for Ian. So you mentioned that AbPro's antibodies are more natural and less toxic than other antibodies. Can you elaborate on this a bit? Speak on this a little bit more.
Sure. Yeah, so the way antibodies have evolved are really now toward there used to be humanized antibodies, but now there are ways to isolate antibodies directly from humans. So they're what we call real human antibodies. So those are the ones that our human immune system have created, and obviously they've been in man already by default. So we already know that those are going to be the safest ones and the most natural.
So one more for you, Ian, and then I have a few also for Ruben. How are these bispecific antibodies poised to have a longer half-life?
Yeah, so through engineering with our platform, there's ways to really make them. The original bispecifics, I would say the first wave, didn't have a full antibody framework. So the way the field has evolved is really to move toward a full natural framework with the antibody, which includes the FC region. And those will have a much longer circulating half-life by default. So just one analogy is that these antibodies are generally they're bigger, so they're going to stay in circulation. So it's a heavier antibody that won't be moved out of the human body as quickly. So that can have huge advantages, especially for people don't have to wear a continuous IV pump, for example, which were the first generation of bispecific antibodies.
Nice. So, Ruben, see you there. How many bispecific antibody molecules has the parameter screening data been tested
on? I'm sorry, could you repeat the question again? I'm sorry.
So this is a question about how many bispecific antibody molecules, or how many different molecules, has the parameter screening data been tested on?
For ourselves, we have been more than 10 bispecific antibodies, which is the FC-containing bispecific antibodies. We have more than 10 projects went through our platform process. So actually accumulated quite some data for those resin screening and also a parameter screening. And this is for now, but as we accumulate more data, we're also looking to build a model that after we do several tests, we can quickly identify the parameters we're going to monitoring. We are trying to build a model that can help us to predict the parameters that we want to adjust. In the same time, we want to build a model that can predict which is the best resin that we can should be used. As we accumulate more data, I think there will be one day we can realize that scenario.
So another one for you about the specific Chinese hamster ovary cell lines that you're using and vector. And there's a question about research license use. Do you have or offer research license use?
It's actually a good question that I can re-elaborate some of our cell line platform information. For the CHOK1 cells we have, we licensed it from ECACC. We have the clear licensed document, so we for sure have those research license with this ECACC, and we ourselves have the GNS and the ADCC both adapted from the ECACC cell line. So if clients reach out to us, you don't really have to talk with ECCC because this is something we already brought out. For us, the only negotiation will be between the client and ProBio. So our client wouldn't have to pay any other loyalty fee or other associated fees to ECACC or other institute.
Excellent. This particular question potentially may be addressed by either of you. So is there any correlation between purity and potency of bispecific antibodies? Looking at purity and potency and what the relationships are, correlation between purity and potency.
Yep.
I don't know if you can address that one. Yeah. Go ahead.
Yeah, I can. Question. For purity, even you have a bad purity, let's say you have byproducts, but your potency a lot of times are still pretty good because potency is not a test that can reflect in your purity problem. The why we want high purity product is more of a concern for safety, because once you have a byproduct, then this byproduct has never been studied, and also byproduct sometimes is a major problem that causes the undesired effect for medical use. So in short, for purity, the major consideration is safety, and most of the time, even you have a low purity or the purity less than 80%, your potency is still going to be good as long as this product have a good function and this product is correctly designed and produced.
Ian, do you have any thoughts on that? Seems up your alley.
Oh, you mean purity and function?
Yeah.
Well, yeah. So, I think generally you want as pure as possible, but as Ruben pointed out, if the bispecific has been designed properly, that's gonna be the most important feature. So if it's been engineered properly with the right features for the right disease, then that's, in our opinion, the most important thing.
Good. All right, let's see what else we've got here. Okay, this one is I think for Ruben. Comparing the traditional monoclonal antibodies, is it more difficult to develop a process for bispecific antibody, and how long a timeline would you expect from DNA synthesis to tox-based release?
For first question, yes. Compared with traditional mAb, it is more difficult to develop a process for bispecific antibody. And so the major energy was put on for purification process development. In terms of cell line, in terms of the cell line development or process development, it is similar to a traditional mAb product. However, for purification development, and also, like I said, the analytical development it is harder and it takes more time. So for ProBio, we already accumulated quite some experience dealing with the bispecific antibodies. I would say for symmetric Fc region containing product, it will take 1 more month compared with traditional mAb. For ProBio, we have 6 months. We can do 6 months from IND to tox batch release. And for bispecific antibody with symmetric structure with Fc region, it will take 7 months.
So not a lot of additional time, I guess. This, here's another one from the audience. So what are the basic differences in the control strategy or specs between the bispecific antibodies with the trispecific and regular antibodies? So this is looking at the control strategy, bispecific versus trispecific and regular.
So I think we can't really divide simply like bispecific antibody and trispecific antibody because a lot of times the control strategy is not really structure-based, but impurity-based. For FC containing a product, like I said, cell line development and the process development are a lot of times are similar to the maps, traditional maps. And for downstream, it's trickier for bispecific antibody or trispecific antibody. How you would control them is really depending on your byproduct you're trying to remove. If some of the byproduct is really tricky and they are similar to our target product, then more like different ways you're going to use. And like I said, if they have a different isoelectric point, then you would use that to distinguish your product in purities. And if they have different size, molecular weight, you may use a different strategy. In summary, we don't really have a control strategy that can divide specific for bispecific antibody or trispecific antibody, because during the process development, what you're trying to remove or what the byproduct is you're trying to move is more important, and those are the ones that actually drive the downstream parameter optimization.
So do you have any recommendations for unstable product CMC?
Oh, yes. For unstable product, for example, if you have an enzyme product, which is very easy to degrade during the cell culture environment, then you may want to consider perfusion process. You can do your perfusion after the product is expressed out of the cell. It gets immediately, like within 24 hours, removed out of the bioreactor, and for the storing tank, you can keep them at low temperature to keep the product stable. And you can also consider to use low temperature purification. You can do your entire purification process in a cold room in order to keep the product stable. And for sure, for the formulation development, you want to consider lyophilization. And for scaling up, it's also a good big part to consider for unstable products. During the downstream process development, more intermediate stability studies should be done in order to guarantee the scale up of the unstable product process.
Awesome. I have one for Ian, and then we'll have one final wrap-up question here after this. You had mentioned the higher yields. What type of yields are you getting now, and what are you kind of seeing as you continue on in terms of your yields?
Yeah, we're finding that our bispecific platform, because of the symmetry of the molecule, you're not going to get close to a monoclonal yield, but you can come potentially quite close to it. So it's going to be probably 80% of what you would typically see in a monoclonal antibody, because of the way the antibody has been put together.
Excellent. Okay, we've got a couple minutes, and so I want to ask one wrap-up question to both of you. We can start with, I guess we can start with Ian. So as you look at the technologies that you're working with right now, and you look at right now and maybe 5 years out and then 10 years out, right? What do you find the most exciting? What's going to develop next? Kind of what gets you up in the morning about what you're working on?
Yeah, no, I think the whole field of antibodies is super exciting. I mean, I think we're now in a next wave of new technologies. We see a lot of real natural antibodies continuing in the field, so that's going to reduce immunogenicity, make it safer for patients.
Right.
We also see continuing lots of new targets that antibodies can actually treat in immuno-oncology, infectious disease, certainly eye care as well. So there's going to be continually these new advancements are going to keep building up, and then just great promise in this field overall. So that's, I think, what gets me up in the morning is the ability to potentially leverage all these to help patients at the end of the day over the next 10 years. So if we can really make a big difference in these disease areas, I think that will help humanity. So that's incredibly exciting.
Yeah. Excellent. Ruben, do you have any thoughts?
For me, it is definitely inspiring when working at ProBio for process development or deliver all the CMC projects. So for me, most important thing working is I can explore different formats of the protein or antibody drugs. At the beginning, it's in the early years of my work is only mAb, but for now, mAb is like the most classic platform. We can use our bispecific, trispecific, those SCFV connected SCFV containing product. I mean, for me, the opportunity and to explore and to do all the different process development for different platform is really exciting to me, and this is something keep me going. And I really enjoy to working with different formats and also different clients from all over the world.
Well, with that, I think that we have run into time, so I want to thank you both for presenting today and teaching us a little bit about your work and sharing your thoughts on answering these questions with us. And I want to thank you, the audience, for attending this Fierce Biotech webinar and submitting so many great questions. Thank you again to our speakers and GeneScript ProBio for making today's webinar possible. This webinar has been recorded, and you'll be able to access the recording within 24 hours using the same audience link that you were sent earlier. Others that you know can still register and watch the event on demand. And so thank you for joining us, and we look forward to seeing you at future events.
Thank you, Julia.